Let's begin with our webinar. So this is actually not a conventional coaching class as you would have it in your 10th, 12th, etc. or IIT GN stuff. This is actually a research lab. Okay. So ideally the story goes like this, that we are a research laboratory and we have a lot of students for internship with us. These students work very hard and they've got some research papers also. They've presented their work at national, international conferences and have got awards also. Okay, but unfortunately, they're not able to crack these exams like Maharashtra Set or Gujarat Set or NET for that matter, UGC, CSIR-NET, etc. So the opportunities which they should get and people with experience who can serve better to the society were not getting their jobs in these colleges or research centers. Okay, so for my students, I have actually started this endeavor so that they can quickly understand what are the requirements of the exam and prepare themselves accordingly and crack the set or net so that the opportunities are available. So then I was thinking that anyways we are doing a lot of work into the area of education and social entrepreneurship. So why not extend this beyond my students? Okay. So that is why we are conducting this webinar, so that if we are benefiting the society, let's benefit on a wider forefront. Why I'm telling you all this is because you would come with a consistent expectation which are generally attached with your coaching classes, right? So you will get something very different over here and you will not get things which you conventionally get at the coaching classes. So what is it and how do we take it further is for what this webinar is scheduled. So let us take a look at how do we take it forward. So see, cracking MHSCT 2025, particularly in life sciences. So we are conducting the webinar for life sciences as well as psychology. Okay, so net set for psychology also will be considered in near future. The purpose, as I defined clearly, is to empower our postgraduate students to become the next generation facility. A gentle reminder to all of you that the content which I will be showing over you is copyrighted, so please do not reproduce it or take a picture or do a video recording of that. So let's begin with our workshop. Okay, so what is the importance of clearing set? Probably all of you know. I don't want to waste time and emphasize more onto that, but just I want to bring in certain aspects which probably students are forgetting. So what happens is a lot of students attempt their net and set exams probably during the first year of MSc or second year of MSc. Okay, and they take about 4 or 5 attempts to qualify. Okay, because roughly if you want to give a NETCA exam, you need to study for 6 hours daily for approximately 1 year. Then your chances of borderline qualifying NET exist. Okay, and for set also you would require ideally to study for 6 months, maybe probably 6 to 8 hours a day, and you would qualify set. So these are the two things. Okay, but what people forget is they keep on attempting to qualify these exams years after years. Now, here's another approach to look at it. If somebody qualifies at one shot without wasting re-attempts, okay, re-attempts may be learning hota hai, there's no doubt about it. But if you just qualify at one shot, one person qualifies, suppose Mehak qualifies it, and she joins as an assistant professor immediately into one of the college. And other person is attempting maybe year 1, year 2, and about 2.5 years later, he or she qualifies his net set. Okay, now what happens is by that time you have already served your tenure of 2.5 years as an assistant professor. You actually get promoted to associate professor. Okay, so one thing is you have got your job. Second thing is you have completed your experience and you have already completed your probation period. So now you're ready actually with an experience to move to a bigger and a better institute, that is if you wish to. Okay, thirdly, you will also progress from assistant professor to a higher thing, and then finally to a professor maybe after 8 to 10 years depending upon your institutional guidelines and requirements, so I've seen a lot of students who have been applying for these exams year after year, year after year. They really work hard and they're not able to get through that. They're not able to get through that because not because they are not intelligent or not because they are not working hard enough. Okay, it is basically because people have failed to understand what is the premise of the examination and what are they testing in you. Okay, we are not looking at the objective. So when we do research, what is the first thing we do? We first figure out what is our objective, and once we have the objective, then we figure out, has anyone else ever done something like that? If the answer is yes, then what are the methods by which they did it? This is how the scientific method arises, right? So we are not doing that when we are cracking these exams. So this is one point which I wanted to make. Apart from what is the importance of clearing net, this is the important part which you should realize, that a repeated attempts is not in benefit of anyone. Apart from that, it will give you, it's a gateway for your assistant professorship. It will give you admissions to your PhD also. It is recognized by UGC, so you will get a salary of UGC scale. And you can also, once you join as an assistant professor, you can write a research grant in aids and get funds to do your research and also have staff working for you. These are other important features of SET which you and me probably already know about it. Okay, so just let me take a moment. Are there any what do I say? Are there any questions or queries in the chat box? Not yet. Okay, so let me again go back to my presentation in that case. Okay, so we have understood the importance of clearing SET. Now the point is, when I have asked you guys regarding what are your major issues, for in the exam. So most of you have said that you either do not have time. That is the number one option which we have got. And the second point you have said is the syllabus is too wide and too complex to cover. The third important point people have come up is you're not able to remember all aspects of the syllabus, so cracking the exam becomes difficult. So the first three important aspects, I will tell you how we can troubleshoot here in our course itself. Okay, so it's absolutely correct, your assumption that the syllabus is really wide. Okay, there are two aspects of the syllabus. One is the teaching and the research, which is called as paper 1, and then the dedicated life sciences ka thing. Now you also have to understand that people who give life sciences exam come from the background of botany, biotechnology, biochemistry, life sciences, zoology, environmental sciences. All these people are available to give this exam. That is why the syllabus is so wide. That's the first thing. Okay, the primary difference between NET and SET is that in case of NET, you can go, so if there are 13 aspects of paper, for paper 2, the syllabus is actually divided into 13 chapters or 13 sections, I would say. Okay, and there are multiple subsections, as I've shown you as a snapshot over here in chapter 1, which consists of 1 to unit 1.10. Okay, so there are multiple subsections of that. But in case of NET, if you study only 7 units of 13, but you are absolutely thorough with that, okay, there are chances that you will be able to crack that exam. Whereas in terms of SET, you would, and that is why there are huge number of optionals which are there in NET. So you will find in NET there are 75 questions they are asking. Probably they'll ask you to attempt only 25 in section C. So there is more than 50%, more than 60% of optionals which are given to you. That is the framework for NET. For SET, the framework is slightly different. They are expecting you to have a wider syllabus coverage, and probably you should be good at the subjects which you have been studying. So I found out most of the people who have joined here are either from the biotechnology background or microbiology background. So what is expected is the areas of your biotechnology and microbiology you should be good at, and you should be able to cover up the whole of the syllabus. That is required for SET. For NET, if you are good at only at your background and you cover up around 7 to 8 aspects of the 13 sections, you are good to go with that exam. Okay, so that is what about the syllabus. Regarding teaching and research, which is paper 1, I have also found out one common mistake which students do is they spend lot of time investing in teaching and research, and or they spend equal amount of time in teaching and research compared to the paper 2 ka aspect. Now what is happening is the weightage for your paper 2 is higher compared to weightage of your paper 1. I'm not saying that paper 1 is not important, rather paper 1 is easier to score. So someone who's good at paper 1 would require lesser marks in paper 2 to crack it, you know. But the point over here which I'm trying to make is that the weightage of marks is more in paper 2 and you're more familiar to paper 2 because you have been studying it for maybe for past 3 to 4 years. All your bachelor's you have studied your biology, chemistry, and maybe one more optional, and then for your master's you're studying for another 2 years the same aspect of your biological sciences. So you have an experience of about 3 to 5 years of biological sciences, but you have lost the touch of mathematical aptitude, the logical reasoning, data interpretations, critical thinking, associative thinking, etc. So paper 1 becomes a challenge over here. So what we ideally have to do is we have to attempt comfortably paper 1. There are 10 sections of that. There are certain sections which you cannot afford to leave out. Okay, those sections, when you join along with us, we identify and tell you, okay, that these are the sections you need to do, and other sections off and on we try to practice it out. Okay, so that's how. So basically what we do in our course curriculum is that we determine exact focal points according to your background, where you need to focus, what you are comfortable with, and just cover those areas in depth. And other areas we prepare you for the superficial understanding or a foundation understanding rather than superficial, so that on that foundation you will be able to answer those questions. Mind you, it does not indicate that the 13 sections, going into each of the section in extremely high depth and covering it up in all in just plain 6 months is almost next to impossible, unless you have an interaction with us for all the days, every day for 2 hours, which all of you have already stated that you are in lack of time, and the syllabus is extremely wide. So what we will be doing for you is we are going to compress the syllabus for you, identify the key flag points which you need to do, the other important foundation points which you should be aware of, on the basis of which you can crack the exam. Okay, so let's take a look at other aspects. So I've already told you about the background of the exam set, you know. So what do you think, why don't many of the students crack set? Is it easy to crack, or is it difficult to crack? What do you think about it? So you guys can write to me in the chat box. What approach do you think is about the exam? Is it really easy but you're not working hard, or is it really difficult and I'm working hard and I'm not able to get my point out? You know, it's like that jigsaw puzzle, that it's extremely complicated because of the number of topics, and we don't know what to do, and we get disoriented and we are not able to focus and not able to crack. Or is it just straightforward and we are not giving enough of time to crack it? Okay, Jay thinks it is difficult. Any other approach? Okay, there's 2 more person. Okay, Max says it's difficult. Anyone else? Difficult. Okay. Okay, so probably with whatever results are coming up over here, unanimously you guys are saying that ये काफी difficult है. Okay, ऐसे लिख रहे हो आप लोग. So let us try to look at the different aspect of it, mainly because no clarity how to crack it. Okay, so Hale says that. Any other aspect you think why it is difficult? One we have already covered, maybe wider syllabus. Another is there's no clarity how to crack. Okay, fine. So we'll develop the clarity. That aspect will pick up in our presentation right now. Okay, due to lack of sufficient knowledge, fine. Any other aspects? Because whatever you guys are going to put over here, only those aspects I'm going to discuss, assuming that those are your pain points, you know, because my work over here is to get solutions for your pain points. Okay, so let's get back to our presentation. Most of you have written that it is a difficult exam. Okay, let's take a look at the result. Okay, so this is the institutional level, whether it's easy or difficult. Okay, Maharashtra set 15th June 2025 result was declared at 5 p.m. and approximately 1 lakh candidates appeared for the exam. Okay, this is the number of candidates who appeared. The number of people who actually fill up the form and don't even go for the exams are much higher than this. So about 90,000 or 1 lakh people appear, and about 6,000 candidates qualify the exam. Okay, that is roughly 6%. Okay, so your CACA result, which has come out recently in Mumbai, or in at least about the Mumbai zone, which I know about, is about 8%. Okay, so it is tougher than your CACA exams. But, and you will also have to think in this aspect that all right, 100 में 6 लोग तो select हो जा रहे हैं ना यार, तो मैं भी हो जाऊंगा उसमें. Okay, it's not like that. It is 1 लाखों में 1 मेरा बच्चा होना चाहिए. आपको लाखों में 1 होना है, तब आपको वो एक सीट मिलेगा, तब आप वो क्वालिफाई होगे, because it is the competition with your peers. Even 1 mark, half mark more than you, the other person qualifies and you are pushed across this boundary. Okay, so the exam is really tough. The syllabus is really wide. It is not a straight walk away. So people who have tried qualifying this exam by taking a leave from their office, maybe 7 days before the exam, and they study day in and day out for 10 days before the exam, 15 days before the exam, it's not going to be so easy to qualify by that mode. Okay, so then how do we go about it? Okay, so this is what we need to understand. How do we go about it? So for that, we have to, for the first level over here, what we understood was the institutional challenge, how the institute sets the paper for you, and how is the challenge, and how is the ecosystem in which you are actually trying to attempt. So this is at an ecological level, you are actually looking at the gamma profile. Okay, now we actually go to the alpha profile, which pertains to you as a student. So at students level, why do you think you're not cracking the exam? So one of the important aspect is there is no structured study plan. There are study plan. You would compare your MSc ka syllabus, something you have studied for your first year, second year, or maybe the third year and first year, second year of MSc, you're good at. So probably you are good at genetic engineering, you'll pick up that. You're good at genetics, so you will do the crosses and the hierarchy and karyotyping related questions over there. Probably you're good at molecular biology, so you will start studying as to the structures of the proteins and the DNA, etc. But what is happening is that is not what the institute is gauging at you. Okay, they are not gauging what you are good at. They're gauging whether you have sufficient. Somebody said they use the word sufficient. Do you have sufficient knowledge to cater to the upcoming students who are going to get their master's certificate? So are you master of the master? That is what they are trying to look at it. And when you're going to conduct your lectures, of course you're going to do a detailed study and then conduct a lecture. So here they want to know that you should be at least a legend in 1 or 2 areas, and you should be a master of multiple trades. Okay, that is why the lack of structured study plan, okay, and the plan which is not dynamic, without a mentorship, does not get you to the results what you want. Okay, there is a second. So what exactly do I mean by structured study plan? There is no learning program for you. You are not putting up as what is important, how this is to be studied, when it has to be studied, what should be the schedule, and how do I plan my schedule. And if my schedule is upset, how do I do the reconciliation? Most of the time either you will leave it, or most of the time you will push your schedule to extreme right, trying to complete your stuff, thereby not completing the important topics. Okay, so that's the major issue, and that's where your mentorship comes into play. Then there is an over dependence on something called as reference books. Yes, even I love my reference books, developmental biology by Gilbert, or cell biology by Baltimore. You just open the books, or Genus Cubica immunology, the colorful images, the lovely diagrams, the classical experiments which they are trying to tell us. You really get so engrossed into that you keep on reading the page maybe for 10-15 minutes. Okay, that is good. That is good as a student. Okay, but that is not good enough to crack the exam. Why? Because even if you buy a normal curriculum from any other classes, any other coaching classes, you will have a big set of your paper one, roughly running up to 800 pages, and then you will have another set of your pages, for your paper 2 and paper 3, which will again run into something like 600 700 pages, so eventually you will end up, even if it will require you to have 520 hours to complete just one set of book, okay, which is not feasible for you because it's going to take 520 hours. You all have already said that you're already in crunch of a time, okay, so you have to be into that zone of concentration and mindset to have so much of time and do these stuff, which you have already said that it is not so easy for you guys. So let us see then how we can do it in other formats. Okay, so we have to concise all the information which is there, which is important. First of all, we have to identify the important from the unimportant stuff, and we have to concise the information, and this information now should be available for you for problem solving. Because if you start reading developmental biology by Gilbert and all these reference books, eventually each of the book, you would know that roughly it runs around 800 pages, so it will be like 16,000 pages, which will take you 2 years of consistent, persistent study, that to 10 hours a day. Then you will have all this information, and that is only plain reading. After that, you will make your own notes. Then you will make another snapshot of that so that you can revise it. So it becomes a poor time management aspect, and that is the major hurdle which students are facing. Okay. Now we go to another point. There's a very weak understanding of paper one as to where to focus and where not, which I've already covered right in the beginning. And the most important point is students either do not get the papers to practice, or they do download the papers, take a printout of it, maybe solve one or two papers, and then there is no consistency in that. You're not doing it consistently. So you just lose the track, and then the exam comes up, and then you just walk into the exam hall thinking that पेपर तो काफी इजी था, मैंने काफी सारी चीजें सॉल्व की, पास तो हो जाना चाहिए. You come with that expectation and it doesn't happen. Why it doesn't happen? Because I just showed you on the preliminary slide that the competition is much difficult than even cracking the CA exam. Okay, so now let's go to the next aspect of it. So the next aspect people said is that of course the syllabus is wide and we don't remember it. So at students level, this is a beautiful Ebbinghaus forgetting curve. So Ebbinghaus was a scientist, he was a psychologist, okay, and he actually did a lot of experiments on learning and memory. Okay, so you must be knowing that Eric Kandel, who is from our field in biological sciences, also did experimentation on learning and memory, and he found out how memory is formed, how transition from short-term memory to long-term memory takes place in A. fulica, that is a giant snail. Which probably, if any one of you are from Sophia or maybe probably taking applied medical sciences, would study about it. So what this curve actually tells you is some interesting points. So this curve actually tells you that if you look onto this aspect of it, this is the percentage of learning which takes place and you memorize or you remember, after reading something and after having after doing a thoughtful learning onto that. So not just plain reading, just reading, probably even writing and probably even drawing notes. तो क्या होता है उसके बाद? So it says that after 19 minutes, 19 minutes after you have read something, you have written something, you have drawn the notes of it, in just flat 20 minutes, 60% of it can be recalled, remaining 40 is lost. You cannot recall it. Okay, that's very natural in all normal human beings. But something dramatical happens when you actually look at day one. Okay, when day one happens, that is, मैंने आज कुछ पढ़ा, okay, and tomorrow I try to recall it. So look at the margin over here, it becomes 35%. So your recall or whatever you have studied comes down to only 35%. That means that 65% of whatever you have studied on day zero, okay, is lost, completely lost. Okay, unless you keep on revising it or unless you keep on working over it, you will not be able to recall that. So we have different strategies and tactics how to work onto these aspects, okay, and have your recall really sharp. Why? Because we have to cover huge amount of syllabus, okay, and we have to recall as much as possible. That is going to give you edge compared to other students. Okay, something which you are good at, probably you will recall it much easier, but other things you will not be able to. So how do we design this? Okay, so one basic problem which you guys do is you try to utilize an overlap of MSc curriculum with the set syllabus. This is the primary fundamental issue which you create, because you take your study का syllabus like अच्छे आपने को DNA के structures है, फिर primary, secondary, tertiary folding of protein है, let's study this, अपने MSc के भी काम में आएगा और अपने set के syllabus में भी काम में आएगा. It doesn't work like that, because set का syllabus is assessing something else, okay, because they're giving you the answers there itself into your question paper, okay, so answers are also given, so they would put things in a very different format, whereas your university ka papers are designed in such a way that you have to articulate what you have learned. So they are going to say that explain the structure of myoglobin, do a comparative analysis between hemoglobin and myoglobin. Okay, so you have different options. You can draw structures, you can write it up, you can do differentiate between two into the pointers. Okay, so that is a different level of assessment which they are doing, and this is a completely different level of assessment which they are doing. So the overlap of the syllabus and studying both at the same time doesn't help you. You have a difficulty in identifying the key topics due to the vast syllabus. We have already spoken about it. Excuse me. And there is lack of exposure to recent question patterns. You know, the question patterns, you must have seen people who have qualified NET or SET probably 5 years back, 2 years back, and then last year. You would find that the difficulty level of the exam is increasing. Okay, and they're asking questions which are up to date. That is, if the new Nobel laureates have been announced and some aspect of their work of maybe protein shuttling is there, they will try to connect that with your current syllabus which is given in your set and net, and also connect it with the methodology ka part. So because we are not reading or we don't have the time frame to do in-depth research or wide review of all these parts, we are not exposed to these things. So when most of the time I've seen students are extremely motivated then when they start with it, as soon as they come with peer-to-peer competition, their motivation drops down. अरे यार उसका इतना हो गया, अरे यार ऐसा हो गया, अरे यार वैसा हो गया, and the consistency is lost. When the consistency is lost, qualifying the exam actually becomes a very big challenge. And there are good institutes which are coaching you for this in Bangalore, in Hyderabad and Delhi, etc. You generally don't find a really good coaching institute all across Mumbai. I have not come across it. Okay, but whatever these good institutes which exist in Bangalore and Delhi, they're charging you like about half a lakh or 60,000 rupees per student for an online, and maybe 75,000 or 80,000 for a in classroom coaching, you know, and they are also coaching students for at least 3 times a week, and Sundays questions, etc. The question paper, mock solving stuffs, etc. So that's how they are doing it. Okay, that's how what is there in the market. So what are the essentials which are required for the exam? So before I move to that, let me just take a look as to if there are any questions. You guys can put in your questions. If whatever we have discussed until now you have any queries onto that, we will sort it out. So I'm giving you just a minute to punch into your questions into the chat box. If you have anything, please put into the chat box. I would be more than happy to take your questions over there. Okay, so what are the essentials, or what is actually required for you to achieve your objective to this Herculean task of actually completing this one exam? You know, probably you must have paid your fees right from your bachelor's to your master's, I'm just assuming roughly like 20,000 or 30,000 a year. First 3 years for your BSc is like 60,000 gone over there, and for your MSc is another 60 over there, including the practicals and stuff like that. So about like a lakh, 2 lakh, 2.5 lakhs you have already spent completing your bachelor's and master's. And still the professional certificate which is required, which actually enables you to get a job, okay, into either a research centers or either into your academia of teaching and other stuffs, is completely absent in this ecosystem, because probably they're teaching you and they're giving you knowledge, but they're not developing the essential skill sets which are required into the industry. So the first pointer is you need to become an objective oriented and focus onto your objective. Okay, this is what our course, so what I'm showing onto this role is what our course provides you with. Okay, so it will provide you with an objective oriented focus. It will show you the integral tactics which are required to qualify the exam. Okay, so there are certain these are not tricks. I'm not saying that these are tricks. Just say how the planets are arranged right from sun towards the outer part of the Pluto, and then they make some acronym for that and then they memorize it and they do it. I'm not talking about those techniques or those tricks. These are simple tricks, I'm talking about actually tactics which help you understand that amongst enormous amount of written content, how do you focus on what you really need to have to develop a knowledge and make connections so that you understand the concept. Okay, so that brings us to the third point, that what exactly to read and how much to read. So we can read Baltimore for molecular cell biology, we can read Robert Brown for cell biology, okay, we can read Janice Cuby for immunology, but which chapters, which papers, what sections we need to read to have it pertaining to the exam is what we discuss during our weekly meets. And that's how when people start studying, they gauge and they start aggregating the knowledge which is of use, usable knowledge, that is where we go. Then we develop regularity for practice of solving. So what we generally do over here is out of the whole of a week, we have reserved 2 days probably for 1 hour or 1 hour 15 minutes, one session. So you will have one session on a Saturday about 1 hour, and Q&A, taking the Q&A together, maybe 1 hour 15 minutes to the maximum, and another session on a Sunday for 1 hour 15 minutes. Okay, that's it. So just 1 hour 15 minutes of study once in a week or maybe twice in a week, taken Saturday, Sunday together. Okay, and continuing this for upcoming 6 months, that brings you to about 48 sessions. Okay, so of these 48 sessions, if you're doing that along with us, there is a very, very high chance that about 90-95% that you will qualify the exam. Of course, there are limitations to that whatever we are telling you, you need to practice that, you need to do it back in your day-to-day activity, you have to be regular with the classes. Is like once you have come, then you have taken a holiday, then please send me a recording, and then you hear out the recording and then you do it on your own. That will not help you to that extent. What you need to do is, I agree that you guys, probably some of you are working, some of you are doing some internship somewhere, so you don't have the time, but at least you have a time on a Saturday and a Sunday, where Saturday we will discuss the overview features of paper 1, and on a Sunday we actually deep dive into the area of life sciences. So at least 1 hour each of these 2 days, 2 hours weekly if you put up, which is monthly, कितना दे रहे हो आप लोग? सिर्फ 8 घंटा पूरे महीने में. And if you do that for consistently for upcoming 6 months, then you are guaranteed to have a successful result. So that is how we work, that is how we actually conduct our course, and we also make you go through the mock exams, so you come to know where do you stand in the mock exams. So if you're not getting 70% in your mock exams, you're not going to qualify NET set at all. So you get an indicator much earlier whether you're going to qualify or not, and this is a continuous process which you do along with us. That's why regularity, practice, and solving the practical problems which they're asking you in the exams is what we do over here. The most important aspect while you're doing this thing is time management, which we have already discussed. Because the syllabus is vast, you're already engrossed into too many things. You have a very low probably a very low aspect of focusing, because by now we've been working, we're doing so much of research, we're studying. Our focus to study consistently for like about 15 hours, 12 hours has reduced drastically. Probably because of this, even the social media which we are going through, the time span, the attention span, and the focal aspects have reduced drastically. So you have to practice and manage it. Then only these things are possible. And then motivation and the mindset, apart from good guidance. You start with a good motivation, you start with a good mindset, but over time you have lost it. So that's where the guidance come, where we tell you that when you are tired, you can't walk any further, can't run any further, we actually do the hand holding and walk along with you to take you that extra mile every day. So every week, that extra mile which you walk, that takes you towards your objective, and then you become successful. So what are the tactical solutions which we are applying? So as I already told you, that we are not a very conventional, people who are conducting the classes and stuff like that. So I'm basically, here's the point where probably I will take some time to talk about myself. Okay, so you actually will learn things from authentic life science experts or mentors who have not only cleared the exam, but they have an industrial experience. Okay, so which industries I have worked in, and which laboratories I have worked into, and what are my experience sectors, that we will take a look at it in maybe in 1 or 2 slides. So looking at over here, okay, what you will see is that I've already worked into a lot of research institutes, premier research institutes in industry. I also conduct wide workshops across schools and colleges, as to how education has to be practiced and how the skill has to be developed. So one of this image is from the Jai Hind College. I've already given a lot of webinars and talks in like Mitibai, NMIMS, SNDT, and number of other schools and colleges. So our area is, we have already have experience in a very large domain. Particularly, you will need someone who can teach you how to crack this exam, has an understanding of how education and learning take place. And at another set, the person has to be from the area of life sciences and biotechnology, who understands the subject itself as an expert. Okay, so we have a number of faculties, which probably will talk in some time. We conduct it in very small batches, So I don't take more than 15 students, apart from my students who are working in my laboratory over here, where they're doing internship with me, for whom I'm actually conducting this and extending it further to the common people. I'm not going to take more than 15 students. Why I'm not going to take more than 15 or 16 students is because I have to focus on each one of you. I want more number of results, rather than more amount of revenue. I'm not doing this for revenue, right? So you'll come to know in a short time when we come to my profile as to what do we do. So my whole purpose is the 15 students who work with me, all the 15 should be able to at least crack set by the end of our course and curriculum. So I need really small batches. I need really to be in personal touch with each one of them on one-on-one basis, and I have to give them personalized attention. Okay, that's why the number is low. We have a lot of doubt solving sessions, okay, and forums. So all your doubts are expected to be put up when we have suppose today is a Sunday. Today, if we are conducting one of the session and the session is over, you have the doubts probably either today or in the following of the working days of the week. You can put up your doubts into our WhatsApp group itself. A dedicated group for that will be created, so that your doubts are not only your doubts, other students also will learn from your doubts. So there is a peer-to-peer learning in the whole process. कि अरे हाँ इसने अच्छा सोचा, मैंने ये सोचा ही नहीं था, ये भी मेरे दिमाग में था. These type of doubts can be sorted out for all the 15 students. Okay, so this increases again your probability of cracking the exam, because now you have an ecosystem which is moving towards one goal, towards one objective through one trainer, and everyone is focused onto that. So your chances of culminating this experiment with a positive result becomes very, very high. Okay, so we, as I told you, we have 2 days a week as our course. One of the days where we take selected topics and doubt solving sessions, which may be a Saturday for paper 1 and Sunday for paper 2. 1 hour 15 minutes, the maximum time cap. Okay, we generally take a session for 1 hour. We extend it by another 15 minutes if there are too many doubts or there are too many exciting things happening over there. Then we have a performance tracking and feedback reports. So these are not digital reports like how you will find in other classes where they'll give you some tests, and some only digital marks will come, like fail ho gaya, pass ho gaya, you need to study more onto these chapters. No, we actually observe you from your questions which you ask during the sessions, the doubts which you have, the way you are studying, and the mock tests which you are doing. We run feedback reports. That is, I will give you a personalized feedback as to what you're doing, how you're going wrong, what you need to change, and how these changes will actually end up into benefiting you or reducing your effort. Like some people will by heart lot of things. So I've seen some of the students in which probably the electromagnetic radiation ka spectrum jo hai na, so they start by hearting visible ka kitna nanometers hai, फिर radio waves kahan aate, they don't know, ultraviolet ke 3 sections hote hain, wo kaun se hote hain. You pressurize your brain a lot on by hearting stuff. We don't encourage that. We would not encourage anything to by heart. We will actually create lot of syntaxes in such a way that you will remember it automatically. That automatic recall is what we are experts into, and that is what I'm trying to show over here. So what we are going to do is we are going to give you a study plan. You will have a regular class schedule. You will start with it in the beginning. You don't have to undergo the loads of the huge reference books. Okay, whatever you have, you can start with that. There's no need to buy new books, or we are not going to ask you to buy anything new as required. Whatever is required, we can share it with you. If you want, you can buy it. It's optional. Okay, so you start being lean and light without any pressure, and you start moving from being starting point on the month 1 to becoming fair, not good, not bad, then relatively better or good, whatever you are doing at. By the month 5th, you should be actually very good, destined to move towards success. By the end of 5th month, we will be able to get a clear picture whether you're going to qualify it or not. So why are you not going to qualify it? Those corrections and errors, course corrections can be done over there so that you get back to your objective. That is what we do during our feedback reports. I do not give you a detailed notes which are like printed notes jo you get it during your 11th, 10th, 12th and NEET ka exam and stuff, because this exam is not where you have to buy out something and go and articulate and write answers. Here the answers are given to you. You have to develop an ability to think, devise patterns, understand how the patterns are related to one another, and then come to an answer. So we give you a structured, crisp notes. These notes are so crisp. I will show you one of the snapshot of the notes that probably in flat 8 pages, the whole of ecology is over. Okay, the whole ecology ka textbook, like a very good textbook like Odam, maybe of like 200 250 pages. That whole 250 pages are concised down into just flat 4 A3 sheets, and these A3 sheets, once you have understood it, okay, you would be essentially able to solve all the questions which are given in this exam. And these sheets also help you to give you an active recall, because I told you that you're going to lose the memory as day goes. If you're going to read through the books and try to revise it, then you cannot cover the syllabus. If you cover the syllabus, then you will realize that whatever you have studied during the first quarter of the syllabus, you have already forgotten that, and you have to redo the thing. So we actually create crisp notes by which your short-term memory will get converted into a long-term memory and automatic recall will happen. So as soon as you see the question, अरे इसका आंसर तो ये होना चाहिए, Before you read the question, you will have the answer, and then you verify by reading through the question in detail, and that is when you will realize that yahi option sahi hai, and you select the option, and there you go, sail through the exam. Okay, that takes you to the sixth part, which is the success. So actually, who's your trainer, who's coaching you? So this is being conducted by scientists. So I myself, I am a scientist, okay, and we have team of other scientists and professors who work with us. We have other people like Ketan Tamankar, who would be conducting some sessions maybe of mathematics if required. So these are actually called in when you require them. Okay, we have Patil, that is Shankar Patil, who is from Max Planck. Okay, he's an expert into the arenas of bioinformatics, biostatistics. So these other mentors will be called into if there is a requirement, and if we see that yes, the ecology and the interaction between the students is entering into a nice domain and entering into a great details, and you need to know these details, we will have these mentors also coming on board and talking to you guys. So I don't conduct these type of things only on the basis of and being an academician. So the experience is not only in theory, it is in practice also. I am one of the board members for the studies in life sciences like botany, etc. Okay, I am a postdoctoral. I've done my postdoctoral fellowship in education as to how education has to be conducted so that the load on the students is minimal and they are able to solve those, enjoy the whole process of education, and essentially an expert into the area of getting things done from you rather than me doing the things and asking you to buy heart. We will actually do the hand holding with you and ask you to ride the cycle until you're confident enough to ride your own bicycle, and then even give you ideas as to which arenas you should explore and how you should be careful in those things. So as a motivator and an achiever, these are the things which we will provide you when we are doing that. So I'm a person who has an experience in R&D and education for more than 15 years. I've worked with the Tata Institute of Fundamental Research. I'm just assuming that you guys know these logos, symbol, and aware about it. I don't have the time to discuss with you guys right now as to what I've done in detail with each of these institutes, which I would ideally love to. But this is just to give you an idea that, okay, I've worked with the Tata Institute of Fundamental Research. I've worked with the Homi Bhabha Center for Science Education. So how science education has to be conducted so that things are easier. I was an invited faculty to the University Department of Mumbai University. I've worked with the KEM Hospital and NIRRH. I was also invited at the National University of Singapore to do the X-ray crystallization studies. I've worked with the Forest Innovations Division in Israel to actually develop an R&D project for the company, the details of which actually due to certain reasons are restricted. I cannot reveal it to you guys. Okay, I've worked with the America India Foundation, which extensively works onto the area of education and extending the education to students who are pursuing science. Okay, so why this course? I've already given you a brief outline that I have students in my research lab who are doing internship, and these students keep on requesting me to conduct certain sessions like this because they're going to the international conference and still not able to crack this exam. It is for them where I'm initiating this. So we thought that let's extend it out to the whole of the system. So now why join our courses? So our institute versus compared to others. So now let me just be very frank. I'm not conducting it on the behalf of my institute. I'm just doing it as a freelancing on my own, because it's requested by my students. So I'm just lending out 3 hours weekend to those students who really need help and who are really passionate and motivated to crack the exam. So we have something which is within a range of 6 to 12 6,000, and other institutes are putting it at the range of 12 to 20 when it comes to online. And for the on-site courses, I've already told you that it crosses more than 50-60,000. So you're going to learn from a scientist compared to a generic tutors. There's always a difference. You'll get a lot of things which are there on the YouTube, but you yourself know that it is not possible for you to go through all the videos onto the YouTube, which will also help you. Okay, but you don't have that sort of time to finish all the videos from the YouTube, and you will also forget whatever you have just heard. Okay, creating notes from those videos again will become a huge problem. So there are a lot of problems when you work with these generic tutors. I'm going to focus only on my 15 students so that they crack it. So I'm not interested in larger batches. Okay, I have a regular review at the end of the week every week, and test series also. And of course, apart from purely academic focus and guiding you the things, you will also have a great motivation when you doom down due to certain pressures. So we focus only on results and not revenue. So that's our motto. What does our course offers? It will be an online classes. Okay, we have paper one practice. So on your paper one, only certain aspects of the paper one can be taken up. Okay, and certain things like comprehension, you just read a paragraph and you just know what is the right answer. I'm pretty sure you all guys are so smart enough that all of you will get mostly the correct answers for that. If you have any doubts, you can ask and we can sort it out, but that's not what we are going to cater you for. But certain sections of paper one will be taken, 2 or 3 sections will be picked up, and we will majorly focus on paper 2 because the weightage goes over there, and we will be practicing it. So you will have paper 1 probably on a Saturday and questions regarding paper 2. You will have paper 2 actually studying on a Sunday, and maybe questions at the same time relevant to that. Okay, we will provide you with a short summary concept map notes. Okay, so what is a concept map notes? I don't know whether you guys are aware of it or not, but these look very complicated, these look very scary. Okay, but once I take you through these notes, you will know what it makes as a sense out of it, and you can finish large amount of portion in just few minutes. So I'm going to show you one example how it happens. Probably I may not have the time to explain the example to you, but just show you the example. Of course, the timely reviews and revisions will tell you where you stand. Whatever we are doing on a Saturday and a Sunday will be recorded, and these recorded lectures will be provided to you on a request. Okay, the reason I'm telling you this is because I don't want it to be uploaded onto my website or onto a YouTube, which eventually I would have to do, I guess. Okay, because thing is, you've already spent 1 hour with me doing that thing. Now what you need to do is practice about it, read more about it, and solve the question and answers or solve the problems. I don't want you guys to keep on reading, keep on listening and reading through my video over and over again. So that is redundancy. We want to reduce the redundancy. But yes, at certain cases it might happen that the student is not well, is gone out of town, not available to attend this session. For such students, we can provide you with a recorded lecture, and of course you will have a WhatsApp support group 24/7 active, which you have already experienced a bit of it when you all have joined with us. You will have a personal attention, and we will identify the areas where you have difficulty in solving things. Like most of you have already written that biochemistry is a major challenge for you. So what is a challenge for you in biochemistry? Is it the structures, or is it the process flow, or is it how you imagine the molecules? What are the issues have to be identified, and these have to be identified at a personal level, because every person is different and every person will have different issues regarding that. So these have to be sorted during the sessions. So we will support you till you pass. So the point which we are doing over here is the support will exist until you pass, but that doesn't mean that you have taken a break, you did not go for the exam at all, you worked with us for maybe 3 months, 4 months, and then you feel like arey yaar nahi hoga mujhse, you are distracted to something else in your life, and then again you come back maybe on a 4th month, 5th month, and then you're not able to pass that exam. So taking breaks is not allowed, because we are extremely serious and we give a large amount of energy and effort behind every student. So we take only sincere and regular students who are serious about their careers. So if that is it, then we can do it. So you may take a break for anything, it may be anything justified, but then if it has created a break, you will have to go through a recorded lectures, and then essentially you're on your own, you know. So with these limitations, okay, and the strengths which we have, we've actually given you a fair idea as to who we are and what we do. Okay, so conventional written notes or books we don't give, test series books we don't give. I will not give you any offline पर्सनल क्लासेस वन ऑन वन कि सर यहाँ पे आके मिलते हैं, आप हमको आके मिलो, हम लोग सब लोग 4 जन बैठ के ये डिस्कस करते हैं. So everything will be essentially online, and you should be well versed with all these setup, you know. So we don't give refund in midway for like I had a marriage, I had a job, I had a transfer, etc., etc. The recorded lectures are there. We can share it with you. You can learn through those things and try your luck at it. Okay, we don't provide any other study material also, and we consider this as a family. Okay, so personal one-on-one class actually creates something like you are something special, and it keeps on getting iterated, and it eats away my energy and time. Our 15 people together will form a family, and that family of 15 people is nothing but one unit for me. So everyone is treated, I wouldn't say essentially same, because every person is different and they will be met with different treatment, so that you will develop your aspects because you're different. So I have to treat you differently so that all of you get to one of the goal, you know. But personal one-on-one classes, personal one-on-one discussions and query solving will not happen. It will happen in common sessions with all of them together, so all of them will benefit. Because these days I've seen this trend which is increasing, you know, that students will go to classes, they'll go and get some probably Aryan ka book or some good nice notes from these classes, and they wouldn't want to share it with anyone because competition है भाई, मैं उसको दिखा दूंगा वो पास हो गया और मैं फेल हो गया तो. So there's no peer to peer learning happening. You're either isolating yourself amongst yourself. We don't encourage that. We consider all students as a family, and all of them will be family members, so everything will be shared with one another, and that's how we all will learn together. ठीक है. So exemplar मतलब an example कि भाई फिर concept notes मतलब आप क्या कहना चाहते हो सर, आप क्या दोगे. So this is a concept note. Okay, so probably ये आपको स्क्रीन पे बहुत क्लियरली नहीं दिखाई देती होगी, बट लेट मी जस्ट टेल यू, ये जितना नोट पे जितने डिटेल्स डाले हुए हैं हमने, ओके, सो ये डिटेल्स आपको थोड़ा सा कॉम्प्लिकेटेड और बहुत सारा ऐसा कंजस्टेड लगता होगा, बट ऐसे 4 पेजेस, इफ यू जस्ट, देयर आर ओनली 4 पेजेस विच कंप्लीट्स योर इकोलॉजी. ऐसे 4 पन्ने आपने पढ़ लिए, पहले तो इसके ऊपर लेक्चर होगा, वो लेक्चर के बाद ही आपको ये नोट्स समझ में आएंगे. सिर्फ नोट्स देंगे तो आपको समझ में ऐसे भी नहीं आएगा. तो मेरे लेक्चर्स होंगे इसके ऊपर 4, ये 4 लेक्चर्स के बाद आपको ये 4 पन्ने समझ में आएंगे. ये 4 पन्ने समझ में आने के बाद आपका इकोलॉजी खत्म. ये 4 पन्ने अब आपको रिवाइज करने के लिए इट विल इनिशियली टेक 10 minutes, देन आफ्टर फ्यू वीक्स इट विल टेक जस्ट 5 minutes, then it will take 2.5 minutes, and there will be a day that you will finish these 4 pages in just 2 minutes, and your ecology revision is over in just flat 2 minutes. This is the strategy which you require when you are studying sciences like life sciences or biological sciences, so that you can recall the data which is deeply stored into your mind and brain and make connections widely, so that you will be able to do it. So here is an example of biochemistry. I actually wanted to give a demonstration. So हमारे पास और 20 मिनट है जहाँ पे लेट मी सी इफ आई कैन गिव यू दिस डेमोन्स्ट्रेशन. ओके, क्योंकि काफी लोगों ने, आप लोगों ने मुझे कहा था कि biochemistry is the subject which most of you find extremely difficult, and biochemistry is the subject, it is like mathematics, you know, एक बार आपको समझ में आ गया तो you can score full. ओके, बायोकेमिस्ट्री में सबसे डरावना इज एंजाइम काइनेटिक्स, है ना. कुछ लोग यहाँ पे माइक्रो वाले हैं, सो ये उनके लिए भी काफी रेलेवेंट है, दैट्स व्हाई आई पिक्ड अप दिस एग्जांपल. अब ये आप पेपर देखिए, यहाँ पे 11 क्वेश्चंस पूछे हुए हैं महाराष्ट्र सेट के एग्जाम में. ऑल 11 क्वेश्चंस हैव कम फ्रॉम एंजाइम काइनेटिक्स ऑफ बायोकेमिस्ट्री. ठीक है, नाउ लेट अस ट्राई टू डू वन थिंग. अब ये जो सवाल उन्होंने पूछे हैं, some of you may not be able to answer it, or probably you will be able to answer it 2 or 3 out of 11, है ना. रफली 2 या 3 आपको आ जाएंगे through your experience, whatever you have been studying. Now let's do one thing. Let's conduct a short one slide course of enzyme kinetics where you will be able to solve 70% of these questions which I just showed you. ठीक है? So I'll just explain you how biochemistry works. Biochemistry you find it difficult because ये actually chemistry that works in biological system. ये aspect, and because biology is a very interconnected complex physiology, biochemistry becomes more confound with these principles and it becomes very complicated. अब इसको समझना बहुत सिंपल है. बायोकेमिस्ट्री को अगर काइनेटिक्स आपको समझना है तो लुक एट द गिवन ग्राफ. ठीक है, ऑन द लेफ्ट एक्सिस देयर इज कॉन्सेंट्रेशन, ऑन द राइट एक्सिस देयर इज टाइम. द रेड वन ऑफ द रिएक्शन इज ऑफ द रिएक्टेंट्स. माने क्या हो रहा है, एट जीरो टाइम रिएक्टेंट्स इज 100%. मतलब एक टेस्ट ट्यूब है, इसमें मैंने केमिकल डाला है और ये केमिकल विद टाइम चेंज होने वाला है. ठीक है, सो नीचे के एक्स एक्सिस पे टाइम है. सो द रिएक्टेंट्स आर गोइंग टू गो डाउन विथ रिस्पेक्ट टू टाइम. खत्म हो जाएंगे ना वो लोग, जैसे जैसे रिएक्शन आगे बढ़ेगा रिएक्टेंट खत्म हो जाएगा और वो प्रोडक्ट बनाएगा. सो दिस रेड लाइन एक्चुअली विल गेट कन्वर्टेड एट द सेम टाइम इनटू द ब्लू लाइन व्हिच इज प्रोडक्ट्स. दिस इज कॉल्ड एज नथिंग बट रेट ऑफ रिएक्शन. ठीक है, इस रेट ऑफ रिएक्शन को अगर आपको स्टडी करना है तो या तो आप ये रेड लाइन के स्लोप को निकालो. ठीक है, स्लोप इज द tangent लाइन विच वी विल ड्रॉ टू दिस रेड लाइन ओवर हियर. ओके, जिसका फॉर्मूला हम लोग ने 10th स्टैंडर्ड में पढ़ा था, y2 minus y1 upon x2 minus x1, है ना. y कोऑर्डिनेट्स के y2 minus y1 upon x2 minus x1 when you draw a straight line tangent to the red axis. Same answer you will get if you draw a tangent to the blue line which is products, because the rate of exhaustion of the reactants is equal to the rate of production. ठीक है, ये समझ में आ गया. This is called as first order reaction in terms of chemistry. As simple as this. Now there is zero order reaction, there is first order reaction, and there is a third order reaction. Let us take a look at it. अब इसके ऊपर बहुत सवाल पूछते हैं ये लोग एग्जाम में. ठीक है, अब आप ये ग्राफ्स देख के डरो नहीं. अब मैं आपको दो चीजें बोलने वाला. फर्स्ट ऑर्डर आपको समझ में आ गया. ठीक है, सो फर्स्ट ऑर्डर के जो ब्लू लाइंस के ग्राफ है वो देखिए. फर्स्ट ऑर्डर का जो ब्लू लाइन का ग्राफ है, इट इज शोइंग यू दैट द रिएक्टेंट्स आर गेटिंग ओवर विथ रिस्पेक्ट टू टाइम, व्हिच इज द रेड कलर का रिएक्टेंट्स का ग्राफ इन द रिएक्शन रेट व्हिच वी सॉ. ठीक है, सो विथ टाइम, विथ इंक्रीज इन टाइम, concentration of your chemical or your reactant will go down. That is the first order. Second order में also it is going down. Okay, second order is only called when there is not one reactant but there are two reactants. It is called a bimolecular reaction. मतलब first order में एक ही chemical है but which is A, whereas in second order there are two chemicals, there are two reactants, A plus B, which gives you C plus D. That is why the curve becomes more deeper, which is a second order. Okay, एंजाइम काइनेटिक्स में आप जब भी कोई भी पढ़ाई करोगे, ठीक है, तो यहाँ पे ना फर्स्ट ऑर्डर रिएक्शन होगा ना सेकंड ऑर्डर रिएक्शन होगा, क्योंकि एंजाइम का जब आपको स्टडी करना है तो आपको रिएक्टेंट्स नहीं देखने हैं. रिएक्टेंट्स आर नथिंग बट सबस्ट्रेट्स फॉर एन एंजाइम. व्हेन यू डू एंजाइम काइनेटिक्स, द इंपॉर्टेंट हीरो ऑफ द स्टोरी इज एंजाइम. Okay, and not the substrate which is a reactant molecule according to the chemistry. So reactant को भूल जाना है आपको और आपको एंजाइम पे फोकस करना है. इसीलिए सारे एंजाइम काइनेटिक्स के रिएक्शन हम लोग हाई कॉन्सेंट्रेशन ऑफ सबस्ट्रेट पे करते हैं, जिसको बोलते हैं सैचुरेशन ऑफ सबस्ट्रेट्स. ठीक है, क्योंकि सैचुरेशन ऑफ सबस्ट्रेट हो जाने के बाद आपको जो भी डेटा मिलेगा, ऑल दैट डेटा यू विल रिसीव विल बी द प्रॉपर्टी ऑफ द एंजाइम इटसेल्फ. समझ में आ गया? सो दैट इज कॉल्ड एज अ जीरो ऑर्डर रिएक्शन फॉर एंजाइम काइनेटिक्स. जस्ट गिव मी अ मोमेंट. Okay, let's get back to this. अच्छा, किसी को कोई डाउट है अभी तक? अदरवाइज लेट अस फिनिश दिस, देन यू गाइस कैन पुट अप योर डाउट्स. ओके, नहीं तो अपना कनेक्ट टूट जाएगा. ठीक है, सॉरी. सो द इंटरेस्टिंग थिंग इज एंजाइम काइनेटिक्स जब भी किया जाएगा ना, So it will be zero order kinetics. So the rate of reaction आपको जब भी निकालना है, that is the first graph over here. अगर आपको मेरा माउस दिख रहा होगा तो okay, तो the rate is always constant. It will be like a flat line. ये आपको देगा properties of your enzyme. ये बाकी के दो जो है is not used in enzyme kinetics. Now let's come to the last part of it, the most difficult जो एग्जाम में पूछते और हमको नहीं आता. ठीक है, KM Vmax वाला. अब ये जो लेफ्ट साइड का मैंने अभी जो ग्राफ डाला है जो हैंड रिटन में है, ठीक है, नाउ एवरीवन कैन फोकस ऑन टू द हैंड रिटन. उसमें दो पार्ट्स है, लेफ्ट ग्राफ एंड अ राइट ग्राफ. ठीक है, द लेफ्ट मोस्ट ग्राफ इज एक्चुअली अ माइकलिस मेंटन इक्वेशन. ये आपको क्या दिखा रहा है? ऑन द एक्स एक्सिस देयर इज अ सबस्ट्रेट कॉन्सेंट्रेशन, ऑन द वाई एक्सिस देयर इज अ वेलोसिटी. तो आपको यही देखना है कि वेलोसिटी मैक्सिमम कब आएगी. जब भी आपकी वेलोसिटी मैक्सिमम होगी उसका आधा वैल्यू आपको km दे देगा. ठीक है, so always remember कि velocity is not dependent on km, but km is half the value of velocity. ये कार्डिनल प्रिंसिपल है, ये आपको अंडरलाइन करके याद रखना है कि वेलोसिटी km पे डिपेंडेंट नहीं रहेगी, हालांकि km will be dependent on velocity. Now what is km? जब आप जीरो ऑर्डर रिएक्शन रन करते हो, जब सबस्ट्रेट सरप्लस अमाउंट में, रिएक्टेंट बहुत सरप्लस अमाउंट में है, तो रिएक्शन किस पे डिपेंड करेगा? ऑन द नंबर ऑफ मॉलिक्यूल्स ऑफ एंजाइम विच इज अवेलेबल एंड द नंबर ऑफ एक्टिव साइट्स विच आर अवेलेबल ऑन द एंजाइम. मोर द नंबर ऑफ एक्टिव साइट्स, फास्टर द रिएक्शन विल हैपन. मोर द नंबर ऑफ एंजाइम मॉलिक्यूल्स, फास्टर द रिएक्शन विल हैपन. दैट इज व्हाई यहाँ पे आपको दिखेगा द ब्लैक लाइन. जितना ज्यादा स्ट्रेट स्लोप उतना फास्ट रिएक्शन, जितना फ्लैट लाइन उतना कम स्पीड ऑफ रिएक्शन. ठीक है, सो ब्लैक लाइन हैज गॉट हाईएस्ट. The blue one is an inhibitor. It is shifting the wave slightly towards the right, and the red one is further down. So as you add inhibitor, ये जाके आपके एक्टिव साइट्स पे बाइंड करेंगे, है ना. अगर मेरे कैटालिटिक एक्टिव साइट्स पे जाके एंजाइम के बाइंड करेंगे तो प्रोडक्ट नहीं बनेगा, तो मेरा एक्टिव साइट ब्लॉक हो गया. तो मेरा रेट ऑफ रिएक्शन, वेलोसिटी ऑफ द रिएक्शन, दैट विल टेक प्लेस बट थोड़ी देर के बाद. दैट इज व्हाई यू सी दैट ऑल दीज थ्री लाइंस आर pointing or reaching the Vmax, but after some time. Same way अगर उसका आप linear आप line Weber-Burke plot करो तो you'll find intersection on the y axis which tells you the velocity. माने the Vmax will not change, the Vmax will be achieved, but it will take longer time. Why? Because the affinity of the enzyme is compromised due to the inhibitors. जब भी आपकी affinity compromise होगी आपका km change होगा. This type of inhibition is called as competitive inhibition, क्योंकि आपके एक्टिव साइट के यहाँ पे आने के लिए कोई दूसरा केमिकल कंपीट कर रहा है. क्योंकि वो कंपीट कर रहा है, रिएक्शन होगा, फुल स्पीड में होगा, बट वो ज्यादा टाइम लेगा. द सिंपल वे टू अंडरस्टैंड दिस इज यू आर ट्राइंग टू स्टडी फॉर योर सेट एग्जाम, बट हर बार या तो घर का काम आता है, या तो कॉलेज का असाइनमेंट आता है, या तो कुछ ना कुछ डिफिकल्टी आती है, बीच में डिस्ट्रैक्शन आते हैं, तो आप कर पाओगे सेट का एग्जाम, बट आपको टाइम लगेगा. ठीक है, और वो टाइम खत्म होने के बाद देन यू विल अचीव योर सेट का एग्जाम्स का ऑब्जेक्टिव. सो इट इज अ कॉम्पिटिटिव इनहिबिशन. ठीक है, आपको compete करना पड़ रहा है आपका टाइम और आपके एनर्जेटिक्स को रन करने के लिए. लेट अस लुक एट नॉन कॉम्पिटिटिव नाउ, इफ यू हैव अंडरस्टूड कॉम्पिटिटिव. अब नॉन कॉम्पिटिटिव में क्या होता है? नॉन कॉम्पिटिटिव में कोई भी आपके एंजाइम साइट के लिए compete नहीं करेगा. ठीक है, बट सो जब वो आपके एंजाइम के साइट के लिए compete नहीं करेगा तो व्हाट इज द मॉरल ऑफ द स्टोरी? km चेंज नहीं होगा. सो लुक एट द माइकलिस मेंटन ओवर हियर ऑन द राइट हैंड साइड व्हिच आई हैव पुट अप राइट नाउ. ठीक है, और ये राइट साइड वाला जो है उसमें यू लुक एट द ब्लैक, ब्लू एंड रेड लाइन. ब्लैक वन इज व्हेन देयर इज नो इनहिबिशन, ब्लू इज व्हेन देयर इज सम अमाउंट ऑफ इनहिबिशन, एंड रेड इज व्हेन द इनहिबिटर इज वेरी हाईली यूज्ड. बट सारे केस में the velocity of the reaction changes. क्यों? क्योंकि now the inhibitor is not competing for the binding site, it is binding somewhere else other than your catalytic site, and it is changing the structure of the enzyme. So the net velocity goes down and your km stays the same. When you look at the line Weber-Burke plot, यहाँ पे Vmax change हो रहा है हमारा, velocity of the reaction change होता है, but 1/km is constant. So यही graphs. Now after looking at these graphs, let us see whether you can solve these sums. Okay, look at this sum now. 11th वाला. I don't know whether you guys can read it or not. let me just read it for you. An experiment was performed to examine the effect of various inhibitors on Km and Vmax. Experimental data was obtained to use to construct a Lineweaver-Burke plot. Which one of the following indicates a non-competitive inhibition? मैंने वर्ड क्या सुना? Non-competitive. जब वो compete नहीं करेगा तो क्या होगा? मेरा Km चेंज नहीं होगा, बट मेरा Vmax चेंज होगा. यहाँ पे Km किसका कांस्टेंट है? यहाँ पे एक ही लाइन है, तो यहाँ पे कॉम्पिटिटर ही नहीं है, तो यहाँ पे बात ही नहीं बनती है. यहाँ पे Km चेंज हो रहा है, ये y x एक्सिस पे Km है, तो यहाँ पे एक Km है, यहाँ पे एक Km है, बट Km तो चेंज नहीं होना चाहिए. यहाँ पे भी दो लाइंस है, बट यहाँ पे दो Km है, तो ये भी नहीं होना चाहिए. द करेक्ट आंसर इज Km, where the Km is constant, which is graph number 11. का आंसर is C, where Km is constant and the Vmax is changing. Now look at all these questions. If you read competitive inhibition, which we studied right now, allosteric enzymes regulate the formulation of the product by competitive, non-competitive, feedback, uncompetitive. Okay, look at this. A non-competitive inhibitor increases Km and Vmax, both decreases this. Okay, then again, abhi ye sigmoidal curve. Sigmoidal curve means only S shape. You will indicate what? Substrate concentration against velocity. ये just किया हमने अभी. Michaelis Menten competitive, non-competitive, cooperative. ठीक है? By which mechanism do you think does an enzyme increase its rate of reaction? ठीक है? उसके बाद में there is another one which is over here. Which of the following is true for uncompetitive inhibitors of enzyme? That means of 11 questions which are kept over here, we just discussed in 5 minutes about enzymes. You can very easily solve these questions. Ideally मेरे को आप लोगों को पूछना चाहिए था कि इसका आंसर क्या है. I just gave you the answer, which is my mistake. I shouldn't have done that. But ये 11 sums जो उन्होंने पूछे हैं, barring the 6th one, which looks slightly difficult in terms of equation, you can easily solve 8 out of these 11, you know, which is 72%. If this is the rate by which you solve your biochemistry, you have actually cracked your set exam. That is how it is going to go, and that is how I am going to take you along with us. So let me have some questions first. Are there any questions? So you guys can either raise your hand, unmute and talk, or you can write into the chat box as question 1, 2, 3, 4. Then we can take the questions and we can close upon to the next slides. So are there any questions? Let me just scroll around onto the chat box and see if somebody's asked it. Okay, किसी को कोई क्वेश्चन है? किसी को कुछ पूछना है अभी तक? Even including this example which we took in biochemistry. So biochemistry is something like mathematics, you know. आप practice करोगे और आपको उसके tactical points पता होगे, how to look at first order, zero order and second order. If you start studying from books like Leninger and all, you will develop the insight, but again it will take very long time. So you need a mentor and a guidance who will take you and accelerate through the process. That is the need of time. अच्छा, तो अगर किसी को कोई सवाल नहीं है तो let me just move to the next slide. Okay. Meanwhile, अगर आपको कोई सवाल है तो आप chat box पे डालते जाओ and someone from the desk will point out to me that there are questions and we can discuss the questions also. So this type of study which I just told you is called as concept active recalling study. ये करने के लिए आपको एक mentor चाहिए, एक group चाहिए and group discussions होना चाहिए, ठीक है? Then you can solve these type of questions, and then it becomes really a cakewalk to crack exams like NET and SET. They are not impossible, they are difficult, but there are tactics and there are strategies and there are people whom you should ring out onto your group to solve these. Okay, so the regular coaching classes who actually conduct classes for these, they have an online course which starts from like 18,000 a minimum. Okay, good classes, really a good coaching class charge you something like 35 to 45,000 for online. and about 60,000 for on-site classroom courses. And they also charge you for the study material separately as an 8,000. Okay. So in our case, our fees, we are not here to generate revenue. We are here to generate results. We are here to bring about transformation into our educational society, because I come from that educational society and see that there are so many troubles amongst our young youth, which I want to help around with, and that is why we are going for it. So our fees is not even 18,000, it is not even 55,000. We are not even giving you in any study material. We are just giving you concept notes and our lectures. You can generate your own study material further from our concept notes. And if you need any study material, we can suggest you, which you can buy from outside. There are a hell lot of good books, which books to refer, which chapters to refer, which sections to refer, and which only pages you should read, and no need to read the whole book. That we will tell you in our lectures and talks. वो हम आपको बताएंगे. Our fees is only 6950 if you register today. It is a one-time full fees for coming 6 months until March. So ये है हमारा almost December. Our first scheduled class begins tomorrow. So the whole of January, February, March, April, May, and probably in May end or May mid, you will have your exams, and that's where it gets over. So our fees is only 6,950 if you register today. The scan code is given over here. Let me also put in some insights regarding these fees. Okay, so ये 6950 हम लोग 6 महीने के लिए चार्ज कर रहे हैं. It will cost you around like 1,000 rupees per month. ठीक है, पूरे महीने का 1,000 रुपया. और ये पूरे महीने में हम लोग 2 सेशंस लेंगे आपके हफ्ते में. ऐसे 4 हफ्ते रहेंगे, मतलब 8 सेशंस होंगे अपने. If you look at these 8 sessions and divide it per session, it is just 145 rupees for session. That means आपका probably एक मील, एक ब्रेकफास्ट, एक आउटिंग, या तुम्हारा एक खाली धुरंधर अगर आप देखने जाओ तो भी 300-350 आपके फूक जाएंगे. मैं ये नहीं बोल रहा हूँ कि आप धुरंधर देखने ना जाओ. Probably it's a very good movie and all of us should watch it. But what I'm trying to tell you is this is an investment which is very petty for you to take your career to next level. This exam, if you crack it, brings professionalism to your life. Passing your BSc Life Sciences, BSc Biotechnology, BSc Microbiology, MSc Microbiology with first class, with distinction does not help you. What helps you is, are you NET set qualified? That sets you apart. वही मैंने बोला, लाखों में एक हो क्या, इसका सिग्नेचर वो है, और लाखों में एक आप हमारे साथ काम करते करते बन सकते हो. So if you are interested in working with us, you can pay on the scan code which is given over here. See, the fees for tomorrow onwards will raise to 8,750 if not booked today. I will tell you why so. ठीक है, क्या होता है, I have to focus only to 15 students plus मेरा batch कल ही start होने वाला है. I have already got few students who are there in the batch. There are only few seats which are remaining, which we need to fill up and start tomorrow's batch. So that is why we are putting it के आज के लिए इतना, because once you pay that, we are starting right tomorrow. उसके बाद अगर आप भरोगे ना, तो again I will have to create more webinars and get more number of students and create a new group and then start getting you on board. और उसमें दूसरा दिक्कत ये होएगा कि we would have lost the time which we have a benefit right now. So new year का resolution आप कर सकते हो कि भाई ये साल ये assistant professor बन के मैं निकल रहा हूँ. But if you lose time, then in lesser time we have to again cover the same things, which I may not be really interested, because we should have sufficient time to work, because we are just going to work only 2 hours in a week, which is like trivial, nothing in terms of how to crack the exams. That is why it is an approach कि आज करोगे तो बेहतर है, कल करोगे तो शायद मैं I may not even take a batch. That is the possibility. And if I do take it, it will not be at the same cost, because time change हो गया अपना. In lesser time we have to perform more. Those people who have a problem financially, because last time when I took a webinar, some students wrote to me that right now जब वो webinar सुन रहे हैं, तो they were not prepared with to pay that amount, you know, outrightly, इसलिए उनका छूट गया वो. So if you guys have any financial problem right now, तो आप ये कर सकते हो कि आज रात तक, आई एम नॉट इवन सेइंग कि भाई वो बाकी के लोग आते हैं ना, एक टाइमर लगा देते हैं, नीचे घड़ी लगा देते हैं वहां पे कि अगले 5 मिनट में अगर आप फीस भरोगे तो इतने इतने सीट्स आपको मिलेंगे, उसके बाद ये ऑफर डबल हो जाएगी, ट्रिपल हो जाएगी, जैसे वो कुछ लोग सेल करते हैं ऑनलाइन. आई एम नॉट इंटरेस्टेड इन डूइंग दैट. I am interested in getting only 15 students for my batch. I want to focus only on my 15 students, and I want to get all my 15 students qualify for my set. That is my objective. That is my aim. That's it. So I don't want batch 2, batch 3, batch 4, batch 5 because मैं यहाँ पे क्लास चलाने नहीं बैठा. मैं ये 6 महीने में अगर 15 अच्छे टीचर्स क्रिएट कर पाता हूँ, that is my contribution to the educational department of Maharashtra. ये मेरा एम है. सो इफ एनी वन ऑफ यू हैव एन इशू पेइंग फुल फीस of 6,000, which I told you, 6,950, you can pay 2,000 right now, block your seat, book your seat, and pay remaining 5,000 before 15 January 2026. ठीक है? That will be probably the next batch का सैटरडे संडे का टॉक विच वी टेक. आई विल एक्सप्लेन यू दिस फर्दर. अगर आज आप भर रहे हो तो 6950 आपको भरना है, आपका बैच एंड आपका सीट कंफर्म हो जाएगा. वन टाइम फीस है फॉर 6 months. अगर आज आप सिर्फ 2,000 रुपया भर के रिजर्वेशन कर रहे हो तो अंटिल 15 जनवरी 2026 11:59 PM तक आपको 5,000 रुपया भरना है. अगर आप नहीं भर रहे हो तो आपका 2,000 रुपया हम लोग रिफंड नहीं कर पाएंगे. रादर ये 2,000 रुपया में आपके 8 क्लासेस या 8 सेशंस होते हैं जो आप अटेंड कर पाओगे हमारे साथ. सो 8 सेशंस विल बी लाइक यू कैन वर्क अलोंग विद अस फॉर 2 months एंड सी हाउ थिंग्स आर डूइंग. ओके. और उसके बाद आपको फिर लगा कि नहीं मेरे को तो कंटिन्यू करना है, आई वांट टू पे. एट दैट पॉइंट ऑफ टाइम वी विल नॉट एडमिट यू विद एडिशनल 5,000 that the offer is like 6,950. वो वैलिड नहीं रहेगी. Any payment after 15 January 2026 will be charged at 8,750. So अगर आपने 2,000 पहले भरे हैं तो then आपको रिमेनिंग 6,750 भरने पड़ेंगे if you are paying it after 15 January 2026. ठीक है? So that is what I wanted to tell you guys. Our batch initiates tomorrow on December 21st 2025. हम लोग ने जनरली अभी टाइमिंग ये रखा है कि इट विल बी अर्ली इन द मॉर्निंग, यू नो. सो एक प्रॉब्लम ये है कि अपने घर पे सब लोग बोलते रहते हैं सोता रहता है, काम नहीं कर रहा है, सोती रहती है, काम नहीं करते. एक सैटरडे संडे थोड़ा सो लेते हैं, तो आप सो लो जितना लेना है उतना. बट वी प्रोबेबली विल स्टार्ट विद 7 or 7:30. ओके, लेट कम लेट, बट द सेशंस विल बी इन द मॉर्निंग. 1 hour सेशन इन द मॉर्निंग. देर आर 2 benefits. एक, सुबह सुबह फ्रेश माइंड में आपका पढ़ाई हो रहा है. दूसरा, आप सुबह उठ रहे हो, घर पे भी एक मैसेज जाता है कि यार ये लोग सीरियस हो गए पढ़ाई करने में एंड दे आर सीरियसली स्टडिंग ओवर इट. एंड यू हैव द होल डे टू योर सेल्फ, जो भी आपको करना है, बिकॉज इट्स अ सैटरडे, अ वीकेंड. यू माइट वॉन्ट टू गो आउट, यू वॉन्ट टू do something, you have other plans, etc. So you can plan your days accordingly in that. ठीक है? And regarding batch timings, if anyone has any issues, you can write to us. Right now, we will request you, if anyone wants to take the offer for today, please pay 6,950 by scanning the code which is given over here. Once you scan the code, please verify the UPI ID that it shows Sinopharm Research Center or Rutvik Thengodkar, and the bank name is DNS Bank. If you pay for today one time full fees, you can pay 6,950. Take a snapshot of the payment and DM to us on the number 9869 345 605. Okay. If you want to only reserve the seat, you pay 2,000, take the snapshot and DM to me on the given number and say when you are going to pay the remaining fees. Okay. So I'll just leave the screen over here and wait for you guys. If you have any question and answers or queries, let me take those. Okay, so Ganpat says, I have completed the payment. There's some other also who have said that I have done the payment. All right. So once you have done the payment, just please take the snapshot and push it across on personally, not in the group. Or personally, you can put it up. May I know from 7 a.m. till what time? It will be for 1 hour, Seema. Okay, so it can be either 7 to 8 or it can be 7:30 to 8:30. So Anubhuti is all, yeah, Anubhuti, we have already received your payment. Thank you so much. Yeah, so Seema, it is like 7 or 7:30. If there is really any trouble with any candidate, we can consider some minor adjustments. Okay. Please share the number on which I should DM the screenshot. Yeah, the number is same 9869345605, which is our group ID number, but just DM it personally. Don't put it onto the group. So if anyone else has any queries regarding anything of these, you can put it here right now. I'm here for another maybe 5 minutes. Can you take an evening batch, Anuprita? Anuprita, I will have to see as to how many candidates ask for an evening batch, because our general observation is that when you put an evening batch, most of the people have some ongoing continuity in the work which is coming in the way, and they don't have a fresh mind to do it. So if you have any particular issues regarding a morning batch, we can work it out. You can, okay, you're working. So you're working on a Saturday also? Okay, so then what we will do is, in your case, you can text it to me. You can definitely attend the Sunday batch, right? Sunday biology wala main paper, which has got about 70% weightage. That you definitely can attend. And for the Saturday batch, let me look up my schedule, and there are few students who are asking for other than morning timings. Maybe I can create a group of few people like you, and take it up. One of it maybe on the evening of a Saturday, or maybe Sunday continuous. That is 7 to 8 first with this batch as biology, and then 8 to 9 paper 1, which we have done with other students on a Saturday, with you on a Sunday. Okay, so you can have both paper 1 and paper 2 on one Sunday continuously for 2 hours if you are working on a Saturday. So that can be arranged. We can work out for you. So if it works out for you, you can scan code and book your reservation today itself, so that we can block your seats. Because the problem is, some of you have already paid into this batch, and we are going to take only 15 students, and if there are 1 or 2 students who's coming after that, I'll again have to take a webinar and again arrange stuff. And I don't want to do this as a full-time job, because I have my own research lab. We have our own research papers to be published. We have our own R&D things which we have to do. So this is just like an additional work, since I have a passion for teaching and we want to help our students. That's how we want to do it for you. So that's what it is. So, and if anyone else thinks that you can bring more students, there is also an offer that every student you bring in for our study group, because the wider and the more diverse the group is, the better study happens. That is what I've seen in terms of education. Okay, so if you're going to get more new students through your reference, so every reference we will actually transfer 10% of discount to your fees which is already paid. Okay, so like if you get 4 students more, you will get a 40% discount, but that is valid only if there are seats remaining. If the 15 seats have already been full, then probably those new references which you have brought may not get a chance. Okay, so they might have to wait until the next year. And another important thing which I forgot to mention to you guys is, NET exam comes every 6 months, but SET is one of the exam which comes once in a year. And it is an easier exam compared to NET. If you are prepared, you are definitely going to crack SET. If you are prepared, you are actually competing at a national level for NET. The chances and the type of skill sets they are evaluating may not allow you to pass. So even though NET is happening with a higher frequency of every 6 months, if you don't crack it, you are losing months after months. But if you study for 6 months along with us, you will be able to crack SET exam with regular studies, attending all our lectures, doing your homework and solving the papers. For sure, you will be able to breeze through the exam by May or June, and you can get back to your career and grow yourself into that career. Okay, so I think we've waited for about 5 minutes and I don't see many questions into the chat box. Let me see once again. Desk, if there are any questions which I am missing on, or if there is any other information which I should provide and I have not provided, please put something onto the chat box so that I will just cover it up. So English, Hindi would be better. Hindi, it's visible, it's difficult for me to study. Okay, tried many times, haven't qualified. I have not planned much, but not about the study, not able to remember multiple things at once, so vast syllabus. Yeah, don't worry about syllabus. The beauty of biology is the syllabus is so vast, और ये वास्ट सिलेबस को कैसे वायर इन करके, जैसे डीएनए इटसेल्फ हैज वास्ट अमाउंट ऑफ इंफॉर्मेशन, बट इसको सही तरीके से पैकेज करोगे, सही तरीके से फोल्ड करोगे और सही जगह पे से उसको ओपन करके ट्रांसक्राइब और ट्रांसलेट करोगे, तो थिंग्स आर गोइंग टू वर्क फॉर यू, कोई डाउट ही नहीं है उसमें. Shine says, yes, we need good strategy and right resource. That is what we are trying to give. Now, Shine, not only good strategy and right resources, you need good सिंसियर स्टूडेंट्स, क्योंकि आज ये सोशल मीडिया के वजह से द अटेंशन स्पैन इज बिकम सो लेस दैट 1 घंटा के 40 मिनट भी एक जगह पे ध्यान रख के पढ़ाई करने के लिए इट्स बिकम वेरी डिफिकल्ट. देयर आर लॉट ऑफ स्टूडेंट जो मेरे को आके बोलते हैं कि नेट या सेट का एग्जाम जब वो देते हैं 3 घंटा एग्जाम में वो बैठ ही नहीं सकते, स्क्रीन को स्टेयर नहीं कर सकते 3 घंटा. 1 घंटा उन्हें उठने के बाद वो निकलते हैं, पानी पीते हैं, टॉयलेट जाते हैं. हालांकि टॉयलेट जाना, पानी पीना अलाउड है, बट टॉयलेट जाना यू कैन नॉट लीव योर डेस्क. दैट इज व्हाट इज द रूल, बट मोस्ट ऑफ पीपल आर डूइंग इट. So yes, it's not tricks, it is tactics, and yes, these are great concepts. So that is what ये सिर्फ एग्जाम के लिए हम लोग आपको ट्रेन नहीं कर रहे. Those people who are in your maybe first year of MSc, आप ट्राई करोगे तो आपका MSc Part 2 के एग्जाम्स में you will get fantastic results, and the pressure, the thing which you worry about कि मैं भूल जाऊंगा because of the vast syllabus, all that will go away. You will start enjoying the study. That is the main point which we have to do. Okay, 2 students. Okay, so 2 have paid full money. Sir, can you please zoom the QR code? Okay, so the QR code is not seen or what, so let me just zoom it. I will also put the QR code onto our group right now after we conclude this meeting, so that अगर किसी को, देखो आप आज मिडनाइट तक आपका 2000 भर के अगर आप ब्लॉक कर रहे हो, आपके सीट को बुक कर रहे हो, तो देन बाय 15th of January you can pay the remaining thing. इस वजह से I will put the scan code also onto our group once we end the meeting, कि अगर भाई किसी को मम्मी को पूछना है, पापा को पूछना है, पैसा वहाँ से अरेंज करना है, तो you can do it. Pay only 2000, start attending the lectures, start working with us, and then before the 15th January you pay the remaining amount. And अगर आपको किसी को genuinely अगर financial problem है, तो you can write to us also. And one alternative I have already given to you, that every student you will bring will confer 10% discount to whatever you are being charged. We will transfer 10% amount back to the same bank account from where you have given us. So yes, I have zoomed onto the QR code for others to take a picture and scan. Okay, Ganpat says I have given the payment, completed payment, I have paid the payment. Seema says timing का मैंने सीमा का बात कर लिया. 7 AM Anubhuti, I have paid, I know that. Seema, please share the number on which I should I should DM the screenshot of payment. या वो shared है. Can you take evening batches? इसका जवाब दे दिया. Same, I am also working on Saturdays. Yeah, so I have already clarified it that those people who are working on Saturdays can definitely attend our Sunday batch 7 to 8 am. Those people who lose their batch of Saturday 7 to 8 will be readjusted on a Sunday from 8 to 9. Okay, or any other convenient time which is convenient popularly for all the participants which are there in the group. Okay, that we can discuss amongst each other and do it after you block your seat. Rare opportunity to get your guidance. So many of us were waiting for. Okay, thank you so much. So let me thank all of you for your time and patience to listen out to me. Wishing you all the best in your life, in your career, and giving this exam. All the best. See you soon. Bye.